Welcome to SNN Network Summer Virtual Event. We are pleased to introduce the next presenter for today's event, Peter Alton, President and CEO of BioCardia. Peter, the floor is yours.
Thank you, Rashawn. Hopefully all can see the slides that are here before us. So BioCardia is a company that's advancing cell-derived therapies for cardiovascular and respiratory disease, and I think you'll find it very interesting how we're bringing personalized medicine to cell therapy for heart disease in particular. So first, our standard forward-looking statements here on slide 2. So the BioCardia story is fundamentally encapsulated in this single slide 3. And the value proposition that we bring to patients is detailed here, as well as the value proposition for investors. We have a very significant pipeline as a very small company. And I note that the 4 programs we're currently advancing into the clinic are entirely BioCardia programs. So the first 2 programs we're advancing are based on the same platform, autologous cell therapy derived from the bone marrow. Both of those programs are intended to provide the primary data and support for the 2 indications that they're pursuing. The first is a setting of ischemic heart failure, which is an enormous unmet need, and the second is for chronic myocardial ischemia. Again, an enormous unmet need. These 2 programs have significant milestones reading out this year. It's the same therapy being advanced, and I'll describe that therapy in some detail. The second platform we have is our neurokinin-1 receptor positive mesenchymal stem cell platform. And this is an allogenic platform that we are advancing for the cardiac patients that are not eligible for our lead program, as well as for acute respiratory distress, our first pulmonary indication. And our third enabling platform is really our delivery system, our Helix platform that we use in our first 3 programs. And that Helix platform is currently under clinical use by 2 partners and preclinical use by a number of other partners. And from that, we develop intellectual property and other value propositions for our broader business. But we also are enabling other leading therapies at select companies to be advanced for cardiovascular indications. Now, I note that the first 2 programs you'll see on the very right of this slide are funded by Medicare. That's somewhat unusual, but what it means is that Medicare covers patient screening costs, they cover patient treatment costs, they cover the cost of investigational product as we invoice them at the centers, and they cover standard of care follow-up, making this a very capital efficient program for BioCardia today. I also note that the Helix programs are funded by partners. So those are all being pursued in a cash flow positive setting. And so as I take this forward, our cash position as a small company, shown here on slide 4, is relatively solid. We have a pretty solid runway that'll take us out into Q4 2022. Our burn rate is roughly $10 million a year or $1 million a month. So this shows you where we've been. We have no debt, and we're aggressively chasing the clinical trials that we are pursuing today. Here on slide 5, you see our primary indication of ischemic etiology heart failure. And it's characterized by a large dilated heart, which has lost its pumping capability. It's more of a spherical heart than a football-shaped heart. And that change in geometry is a result of progression of heart disease over time. This is a poster child of unmet clinical need worldwide. Fundamentally, there are no real therapies that enhance a patient in heart failure. Almost all the therapies that are available, and I think literally all of them that have been available in the last 20 years, slow the progression in heart failure. And so they don't reverse the course of the disease. And as I'll share some data with you, there's potential that we will actually, in fact, change the course of the disease. So the market in heart failure is enormous. I note that we are focused on a subset of that market, essentially roughly 1.7 patients who have ischemic etiology, New York Heart Association 2 and 3 heart failure. These are patients who have significant disease. They're not just beginning disease progression, and they're not at end-stage disease where they have comorbidities. And the reason for that is, one, it's the broadest indication. It's a broad indication. And second, it is the patients who are most likely to have a high signal-to-noise ratio in the clinical trial, because the objective here, upon success, is to have a product that's worthy of registration in the United States. I note that we're only going to be treating 1.2 million of those 1.7 million patients, and that's because we have a proprietary cell selection approach that looks at the potency of cells in these patients to make sure that they're suitable for receiving this autologous therapy. And again, this is a therapy from a patient's own marrow delivered to their heart. There's enormous data supporting this, shown on slide 6. These are the cells that normally home to the heart in injury. And what we're doing is we're essentially amplifying this effect by delivering these cells in a very high concentration around damages in the heart in a more chronic setting. There's a couple of comments here on the specific mechanisms of action, and there's enormous data supporting that these cells facilitate angiogenesis, reduce local inflammation, have an impact on oxidative stress and apoptosis. And fundamentally, the scientific literature these days is full of explanations on why this works. And there's general consensus that there is benefit here. What we're trying to do is demonstrate in a clinical trial that this has sufficient benefit for product registration. And so as we're taking this forward, what we've done is we've really done everything we can to optimize and control the therapy from a signal-to-noise ratio perspective and make it so that this therapy fits within the armamentarium of the physicians who are treating these patients. And so what we have here is we've developed a therapy that involves a pre-procedure screening assay, as you see on the top left here of slide 7. And that's a proprietary assay that essentially only allows the patients to advance to therapy if they have appropriate cells for the therapy. Essentially, it's assessing the characteristics of the therapy itself before a patient's allowed to enroll in the clinical trial. Now, that does limit our patient population in the trial significantly, but what it also does is it maximizes our probability of technical success. And I note that this pre-procedure screening assay and the way we are advancing this therapy solves the two primary problems associated with autologous cell-based therapies beyond the issue of efficacy. The first is patient-to-patient variation. You can view this pre-procedure cell potency assay as a quality control on patient-to-patient variation of their own autologous cells. The second thing it does is it results in a collapse of the cost of this therapy. Today, autologous cell therapies for other indications in immuno-oncology are priced in the $500,000 range. And by doing this with a pre-procedure assay and not a remote manufacturing of these cells, we can provide a therapy that's very cost-effective, both for the patients but the healthcare system more broadly. And once a patient qualifies for therapy, we have a procedure that's all done in a single setting in the cardiac catheterization suite using standard techniques and no significant capital equipment, in which we harvest cells from the hip bone, which is the iliac crest, and we take about 60 cc's, which is about 4 tablespoons, and then the cells are then processed right there at point of care in an FDA-cleared platform that we have, and then the cells are delivered right after that 20-minute cell processing with a catheter-based system that we've developed, which is approved in Europe. And today the patient is kept overnight, but ultimately we anticipate they'll be discharged the same day. And it'll be a very straightforward therapy to advance. Again, I note that this is regulated by the Food and Drug Administration, the Center for Biologics Evaluation and Research, but it's regulated as a device system. And Medicare is reimbursing us, as I described, for the 2 lead clinical indications we're advancing. So I'd like to dive in a little bit more detail here on, what is it that we're really doing? And what we're doing is we're optimizing this therapy with a comprehensive approach. We've very much dialed in on patient selection, inventing new diagnostic technologies, which have issued patents in the United States and Europe. We're focusing on delivering a high dosage of cells, which we obtain with a state-of-the-art point-of-care cell processing platform, and that's magnified by our use of a very efficient delivery system, which also has a reported safety profile that's second to none in the scientific literature. And we're bringing these together with an approach that can be low cost, that enables us to go after literally the full 1.2 million patients in the United States that we feel that this is suitable for. So the next slide here on slide 9 details the cell selection potency assay. And fundamentally what we're doing is we're characterizing these cells before the procedure. We're sending them to a core laboratory that is a CLIA-CAP laboratory, and we're saying, does this patient have the appropriate biomarker signature of their cells for us to advance them to therapy with the confidence that they have a very high probability of being responsive to therapy? One of the markers in the biomarker panel we're using is CD34. We haven't been public about the other markers and approaches we're using, because just detailing that we are using CD34 as a pre-selection criteria for these patients resonates with all the KOLs in this field. The other markers are based on previous clinical data that we have, as well as the data in the literature. And in fact, with this cell potency assay, I don't know of a single clinical trial that has been performed that doesn't support that we should see efficacy from our own clinical trials, including our own phase 1 and phase 2 studies. This is truly a personalized approach to medicine. Not only do we select the right therapy for the right patients at the right time, but the therapy itself is derived from the patient's own tissues. And this resonates for physicians, for patients who are keen on knowing that they're likely to be responsive to therapy in advance and know that there's a much lower risk associated with an autologous therapy, which is unlikely to cause an immune response. It also resonates with payers, because payers view this essentially as we're focusing the therapy dollars on the 70% who are most likely to respond. Once a patient's selected, they enter the harvest, cell processing, and treatment cycle. And cell processing is unique in that it's done with a platform technology that's also proprietary to BioCardia, in which we mix no reagents with the autologous tissue. Essentially, we have a very elegant, density-tuned buoy system here, in which the bone marrow, which appears as blood, as you can see in this left image, is processed in this cartridge in a centrifuge, and it isolates out what you see, this arrow here on the right-hand side, of what we call the autologous bone marrow mononuclear cell layer. And that's done over a period of 15 minutes in a centrifuge at 3,200 revolutions per minute. This provides us with a 5 cc dosage form with a target dosage of 200 million cells that we then deliver to 10 regions around the damage zone in the heart. And these cells contain all of the cells of greatest interest for cardiac cell therapy over the years. I note that this platform is approved in the United States, it's approved in Europe under CE mark, and it's approved in Japan for therapeutic purposes. The approval in the United States is not yet for therapeutic purposes, and that's one of the things we hope to deliver as a result of this clinical trial. So the delivery of these cells then takes place immediately after. So while the cells are being processed, the patient is positioned for cardiac catheterization, and catheters are inserted into an artery in their leg and advance up and over the aortic arch, as you see here on the right, and into the chamber of the heart, the pumping chamber. And from within that chamber, this catheter system has a corkscrew needle that screws into the myocardium and is stable in the myocardium. And this catheter system is what we view as best in class worldwide today. It is approved in Europe, and it's been used in many clinical trials. To understand how it works, hopefully this animation will work, we have an image here of how the cells look when delivered into the heart. And what you're seeing here is contrast media delivered intramyocardially in a swine heart. And so this is what the dosage would look like for a single depot of cells into the heart tissue. And so, as you can see, we can deliver cells locally into the heart. Now, if the same material was delivered into the coronary artery, it would flush straight through the heart. And so the delivery is an important element that enhances the efficiency of our cell dosing. Now, I have a lot of complicated slides to share that are based on the clinical data we have available today. And I'll try to go through them quickly and share primarily the high-level comments. So here we're detailing our phase 1 work in an open-label clinical trial. This means that both the patient and the physician knew that the patient was being treated. And across all metrics, patients performed well. And there were no safety issues, treatment emergent major adverse cardiac events in any of the patients enrolled. I point you to this bottom graphic, and I know it's small. What you're seeing here in the red line, this is all of the patients treated. Their survival out to 3 years in heart failure versus historical controls in green. And I note at 3-year follow-up, there's roughly a 30% reduction in mortality related to historical controls, in addition to showing improvements in other metrics. And this was the first trial of its type, focusing in on patients who had ischemic origins for their disease, but they were not actively ischemic. And we have broad patent coverage around that approach. And based on the excitement from this work, we initiated a phase 2 trial. And this phase 2 work was a placebo-controlled randomized trial, one of the most rigorous clinical trials you can do. It's a smaller trial of 34 patients in this study. And what we see is patients, this is the totality of data, at 1-year follow-up, you're seeing that the patients met the primary endpoint, and in the treatment group, there were no treatment emergent serious adverse event. And actually, in the placebo group, there were no treatment emergent major adverse cardiac events, showing that the delivery that was used in both has great safety support from this trial as well. I note that there were no major adverse cardiac events in the treatment group at 1-year follow-up, although there were at least one in the patients who were in the control group, a 10% profile versus a 0% profile in the treatment group. And what we see here is that all of the other pre-specified secondary endpoints are favoring therapy, where this check mark is. These are respected endpoints in cardiac clinical trials. And what's remarkable is the first 2 endpoints actually were statistically significant in this clinical trial. So based on the excitement for this clinical program, we initiated our phase 3 program, which is currently enrolling today. And this is shown here. There was a 10-patient rolling cohort completed, which I'll share some of the results on shortly. But fundamentally, this is a 260-patient trial. We've moved into the New York Heart Association class 2 and 3 patients to maximize the signal to noise. And I note that this endpoint, when discussed with the FDA, they said that if we move to this endpoint, they could consider this trial for approval, this therapy for approval based on the strengths of this one trial. I note that the power of this trial, which is sort of the statistical probability of success, is such that even if we only see 50% of the benefit that we saw in the phase 2 trial I just shared with you, that this is 90% powered. And what this statement is it's directly taken out of our statistical analysis plan that's part of the FDA submission we are working towards on this trial. There's a number of other pre-specified endpoints. The primary endpoint I just detailed, the Finkelstein-Schoenfeld, is a 3-tier that includes MACE, major adverse cardiac event, mortality, and the functional capacity as measured by 6-minute walk. And in the rolling cohort, we have data that feeds into that. So this is the first 10 patients enrolled in the trial. I will detail, but before I get into that, I'd like to slow and say we just completed a data safety monitoring board review in this trial, and that's where very distinguished cardiologists and biostatisticians get to see all of the data. They look at all of the safety issues. They look at how patients are doing from a risk-benefit equation, and they do essentially a futility analysis. And we recently cleared the latest DSMB review where the DSMB said that the trial should continue as planned. 75% of the patients in this 97-patient DSMB review had reached the primary endpoint in the trial. So we view that as a very big deal, and we're excited on the prospects of our actually having a positive trial ahead, and we're being diligent to deliver the enrollment that this enables. So you can see some of the data we have from the rolling cohort I provided here, and what you'll see in each of these graphics in the bottom right-hand corner, it details the results from phase 2. So you're seeing our 6-minute walk result, how far a patient can walk, is improved over the results we had in phase 2. You're seeing the quality of life that the patients are seeing improved in this trial are also better than we saw in our phase 2 in this rolling cohort. And when we look at the results from our Echo Core Lab at Yale University, we're also seeing that the global left ventricular ejection fraction, or the portion of the blood that's ejected with each stroke, is improved, as is the other wall motion issues. And down here in the bottom of slide 17, you'll see the scientific publications in which support all of this work, and we're happy to provide those. So we also had a publication just recently at the European Society of Cardiology Heart Failure, in which we detailed the dosage form results from the first, I think, 54 patients enrolled. And what you're seeing here is we are delivering essentially the highest effective dosage of CD34 cells relative to 4 previous trials that have demonstrated benefits in these patients. And so other basic science data here also supports our likelihood of being successful in this trial. So the same therapy is being advanced for the indication of chronic myocardial ischemia. And as time is short, I'll advance relatively quickly. We view this as a smaller but still enormous market opportunity, and this is characterized typically by severe debilitating chest pain. And there's no great therapies out there today that are available. Here is the data that's been shown on the CD34 cell front, which is quite compelling, as well as on the left-hand side of this graphic, you see our trial design. Although this is a 343-patient trial, we are initiating. In truth, we're hopeful that this will have an early readout from an adaptive trial design, and we're still working through the specifics of the adaptive statistical analysis plan with the FDA. We've said that this trial will be enrolling shortly this year. First patients have actively been consented, so the press on this program is literally imminent. And for the patients who don't qualify for our lead program, we have our allogenic neurokinin mesenchymal stem cell program we're advancing. These are off-the-shelf cells. Peer companies in this space include Mesoblast and Athersys. The key difference is our cells are neurokinin 1 receptor positive. And so we will likely view ourselves as competitors to them, even as we may wind up partnering with them on the delivery side if they do cardiac programs. What we have here is this is following 3 previous clinical trials we've been involved in and published with culture expanded mesenchymal stem cells. The cells are shown in this image here, and these are manufactured at BioCardia. The current status is we're aiming for IND acceptance in 2021. The previous clinical studies we performed are detailed here, all of which met the primary endpoint on treatment emergent major adverse cardiac event, and there were none. and all of which showed interesting potential benefits associated with these therapies, these mesenchymal cell therapies. So this is an exciting program. I note that the patients are currently being lined up for this therapy today because they're being excluded from our leading program. So we're hopeful that this will have its IND acceptance this year. We're also hopeful that this will have an orphan indication, which it does not yet have, but is something we hope to discuss with the agency ahead. And with that, these three programs, the two autologous, which utilize the equipment shown in the top right image, and the allogenic, which will be shipped in a cryo canister on the bottom right using the same delivery system, all have potential to be pivotal programs. Now, these mesenchymal cells are really compelling. The neurokinin-1 receptor responds to substance P, which is a primary mediator of inflammation in the lungs. And we are advancing these cells also with an intravenous administration to treat respiratory distress that's secondary to COVID. And so this is another significant milestone in the year ahead to have this IND for these cells for the second pulmonary therapy approved by the agency, and we're hopeful to have news in the not too distant future. The intellectual property estate is enormous. There's greater than 65 patents and patent applications that we have worldwide. And the team that's doing this work, as detailed here, are fabulous. All diligent, all capable, and all highly ethical people who have their goal to really help patients from these therapies that we're advancing. So with that, I have a few minutes left. The milestones that we're aiming on for here, I guess I'll take questions. The big milestones are initiating and having first data in the second program and delivering 2 INDs in the third and fourth program this year. So I'm going to take Q&A for the last couple of minutes. I'm looking at it here on my screen. So what are some value catalysts for the company in the rest of 2021? So that's from Robert Kraft. Thank you, Robert. I detailed the 3 in the last slide. And by the way, these slides are available on our corporate website as well. Really, it's starting the second program for CardiAmp in chronic myocardial ischemia and delivering the first safety data set and the 2 separate INDs accepted. You'll also see continued, hopefully accelerated enrollment in the lead program. I have another question here. With heart disease, unfortunately, causing such a large amount of deaths in the U.S. and globally, why hasn't the U.S. FDA fast-tracked the CardiAmp cell therapy? The way to have CardiAmp fast-tracked and enhanced would be through a breakthrough device designation. Stay tuned on that one. And then the last question here is, in your estimate, what length of time will therapy be necessary to begin to show a condition reversal? In the previous data sets we've seen it in 3 months and 6 months and a year. It really depends on the size of the trial to know exactly what's optimal. Our current belief is that the benefit is realized in 3 months and grows a little bit into 6 months and is stable out a year, although we can't prove that out today. I do note that in these rigorous placebo-controlled trials, there's often a very strong placebo effect that lasts out to 6 months based on the literature. So our endpoint in this trial is a 1-year endpoint, primarily to avoid potentially a placebo impact on the signs of efficacy that we need for product registration and ultimately success of the trial. So with that, I really appreciate the questions. I appreciate everybody who's tuned in, and I wish everyone a great conference ahead. Take care. Bye-bye.
Ladies and gentlemen, this does conclude BioCardia presentation. Thank you for joining. You may now disconnect.